Teaming with Microbes, Chapter 3, Bacteria. Bacteria are everywhere. Few gardeners appreciate that they are crucial to the lives of plants, and fewer still have ever taken them into consideration. Yet no other organism has more members in the soil, not even close. In part, this is because these single-celled organisms are so minuscule that anywhere from 250,000 to 500,000 of them can fit inside the period at the end of this sentence. Bacteria were the earliest form of life on Earth, appearing at least 3 billion years ago. They are prokaryotes. Their DNA is contained in a single chromosome that is not enclosed in a nucleus. Their size, or more precisely their lack thereof, must be the main reason our familiarity with bacteria is usually limited to the diseases they cause and the need to wash our hands before eating. Most baby boomers used a standard issue 1,000-power microscope to study microorganisms, but bacteria are too small to see in any detail at this power. School microscopes have gotten better, and some lucky students now do get a closer look, literally, at bacteria. The three basic shapes, all represented in the soil, are coccus, spherical, or oval, bacillus, rod-shaped, and spiral. Bacteria reproduce, for the most part, by single-cell division. That is, one cell divides and makes two cells. They each divide again, and so forth. Amazingly, under laboratory conditions, one solitary bacterium can produce in the vicinity of 5 billion offspring in a mere 12 hours if they have enough food. If all bacteria reproduced at this rate all the time, it would only take a month or so to double the mass of our planet. Fortunately, soil bacteria are limited by natural conditions, predators, protozoa chief among them, and a slower reproductive rate than their laboratory cousins. For example, bacteria must have some form of moisture for the uptake of nutrients and the release of waste. In most cases, moisture is also required for bacteria to move about and to transport the enzymes they use to break down organic matter. When soils become too dry, many soil bacteria go dormant. Bacteria, incidentally, rarely die of old age, but are usually eaten by something else or killed by environmental changes and then consumed by other decomposers, often other bacteria. Primary decomposers. Despite their tiny size, bacteria are among the Earth's primary decomposers of organic matter, second only to fungi. Without them, we would be smothered in our own waste in a matter of months. Bacteria decompose plant and animal matter in order to ingest nitrogen, carbon compounds, and other nutrients. These nutrients are then held immobilized inside the bacteria. They are released, mineralized, only when the bacteria are consumed or otherwise die and are themselves decayed. Different kinds of soil bacteria survive on different food sources, depending on what is available and where they are located. Most, however, do best decomposing young, still fresh plant material, which composters call green material. Green material contains lots of sugars, which are easier for bacteria to digest than the more complex carbon compounds of other plant material. Composters call this brown material, and until it is broken down into smaller carbon chains, other members of the soil food web more readily digest this than do bacteria. Given their diminutive size, bacteria must ingest what are necessarily even more tiny pieces of organic matter. How do they do this? The short answer is they take in food directly through their cell walls, which are composed in part of proteins that assist in this molecular transport. On the inside of a bacterium's cell wall is a mixture of sugars, proteins, carbons, and ions, a rich soup that is out of equilibrium with the less concentrated mixture outside the cell wall. Nature likes to try to keep things balanced. Normally, water would flow from the dilute solution without into the more concentrated one within, a special form of diffusion known as osmosis. But in the case of bacteria, cell walls act as osmotic barriers. Molecular transport across the cellular membrane is accomplished in several ways. In the most important, active transport, the membrane protein act as molecular pumps and use energy to suck or push their target through the cell wall. Nutrients in, waste products out. Different proteins in the membrane transport different kinds of nutrient molecules. One way to imagine this is to think of an old-fashioned fire bucket brigade, in which the water was passed from its source to the fire. These proteins pass buckets of nutrients into the cell. Active transport is a fascinating but complicated process fueled by electrons located on both sides of the membrane surface. The gardener should certainly be aware of and appreciate how bacteria feed, but only needs to understand that bacteria break up organic matter into small, electrically charged pieces and then transport these through their cellular membranes, ready for use. Once inside the bacteria, the nutrients are locked up. Other members of the soil food web obtain their energy and nutrients by eating bacteria. If there aren't sufficient numbers of bacteria in the soil, populations of these members of the soil food web suffer. Bacteria are part of the base of the soil food web pyramid. Feeding bacteria. Root exudates are favorite foods for certain soil bacteria, and as a result, huge populations of them concentrate in the rhizosphere, where bacteria also find nutrition from the cells sloughed off during root tip growth. But not all soil bacteria live in the rhizosphere, for, fortunately, organic matter is almost as ubiquitous as bacteria. All organic matter is made up of large, complex molecules, many of which consist of chains of smaller molecules in repetitive patterns that usually contain carbon. Bacteria are able to break the bonds along certain points of these chains, creating smaller chains of simple sugars and fatty and amino acids. These three groups provide the basic building blocks bacteria need to sustain themselves. Bacteria use enzymes both to break the bonds holding organic chains together and to digest their food. All this is done outside the organism before ingestion. Untold numbers of enzymes are employed by bacteria, who have adapted over the millennia to attack all manner of organic and even inorganic matter. It is an astonishing feat that bacteria can employ enzymes to break down organic matter while at the same time not impacting their own cell membrane. Air and no air. There are two main groups of bacteria. The first, anaerobic bacteria, are able to live in the absence of oxygen. Indeed, most cannot live in its presence. The bacterial genus Clostridium, for example, does not need oxygen to survive and can invade and destroy the inside soft tissue of decaying matter. Byproducts of anaerobic decay include hydrogen sulfide, think rotten eggs, butyric acid, think vomit, ammonia, and vinegar. The notorious Escherichia coli, E. coli, and other bacteria normally found in the mammalian gastrointestinal tract, and thus in poorly made manure-based compost, are facultative anaerobes, meaning they can live in aerobic conditions if they must, but prefer anaerobic environments. Most gardeners have smelled byproducts of anaerobic decomposition, perhaps in the garden, but certainly in the refrigerator. These are smells to remember when composting and gardening with the soil food web, because anaerobic conditions foster pathogenic bacteria and, worse, kill off beneficial aerobic bacteria, the other major group of bacteria, those that require air. While some facultative aerobic bacteria are able to live in anaerobic conditions if they must, most cannot. Aerobic bacteria are not normally known to cause bad smells. In fact, the Actinomycetes of order Actinomycetes, specifically the bacterial genus Streptomyces, produce enzymes that include volatile chemicals that give soil its clean, fresh, earthy aroma. Anyone who has gardened recognizes this smell, the smell of good soil. Actinomycetes are different from other soil bacteria. They actually grow filaments, almost like fungal hyphae. Some scientists believe Streptomyces species use their branching filaments to connect soil particles so they, along with the soil particles, become too big to be eaten by their natural predators, the protozoan ciliates, which would engulf and ingest them. Actinomycetes are particularly adept at decaying cellulose and chitin, two difficult to digest brown carbon compounds, the former found in plant cell walls and the latter in fungal cell walls and in arthropod shells. These are not normal foods of other bacteria. Actinomycetes are also adapted to live in a wider range of pH than other bacteria, from acidic to alkaline. Decay of cellulose. Cellulose, a complex carbohydrate made up of long chains of carbon-based glucose, is the molecular material that gives plants structure. It constitutes half the mass of plant bodies, and hence half the mass of organic matter created by plants. Specialized bacteria, like the aptly named Cellulomonas, carry cellulose-breaking enzymes that they release only when they come into contact with cellulose, as opposed to the random release of enzymes by other bacteria who eat in a hit-and-miss manner. Most bacteria reach their limit when it comes to the non-carbohydrate lignin, another prevalent, molecularly complex plant material. Lignin, the tough brown component of barks and woody materials, is a much more complex organic molecule than cellulose, made up of chains of interlinked alcohols. These are resistant to the enzymes produced by most bacteria and are left for fungi to decay. Element cycling. One way of looking at decay is to view it as nature's recycling system. Bacteria in the soil food web play a crucial role in recycling three of the basic elements needed for life: carbon, sulfur, and nitrogen. For example, CO2, carbon dioxide, is a major byproduct of aerobic bacterial metabolism. Carbon, tied up in plant and animal biomass, is cycled into CO2 gas during decay. Photosynthesis in higher plants converts the CO2 into organic compounds, which are eventually consumed and then recycled back to CO2. Similarly, sulfur is recycled. Sulfur oxidizing bacteria use the element to make plant-available, water-soluble sulfates. Liberated from organic materials by anaerobic bacteria, sulfur-containing compounds are produced by chemoautotrophs, bacteria that get energy from the oxidation of sulfur. The nitrogen cycle, propelled in part by specialized bacteria, is one of the most important systems in the maintenance of terrestrial life. living organisms produce the vital organic compounds, the building blocks of life, amino and nucleic acids, using nitrogen. The strong bonds holding atmospheric nitrogen, N2, molecules together make this nitrogen inert for all practicable purposes and useless for plant needs. For plants to be able to use nitrogen, it has to be fixed, combined with either oxygen or hydrogen, producing ammonium, NH4 positive, nitrate, NO3 negative, or nitrite, NO2 negative, ions. This important process is called nitrogen fixation. Certain bacteria convert nitrogen from the atmosphere into plant-available forms. The genera that accomplish this nitrogen-fixing feat are Azotobacter, Azospirillum, Clostridium, and Rhizobium, any one of which would be a great name for a comic book superhero. Azotobacter, Azospirillum, and Clostridium live free in the soil. Rhizobium species actually live in the root tissues of certain plants, particularly legumes, where they form visible nodules. We don't mean to suggest you need to memorize the species of soil bacteria, but we do want you to focus on the fact that nitrogen fixation, as well as the recycling of carbon and sulfur, requires the interventions of living organisms. These are always taught as chemical processes, but they are really biological. Bacteria carry out these processes in the soil, forming symbiotic relationships with specific plants or existing symbiotically within organisms. Sounds like a case of the biology creating the chemistry to us. Another part of the nitrogen cycle, the place at which it starts in the soil, involves the decomposition of proteins into ammonium, NH4 positive. This ammonium usually figures as part of the waste product produced by protozoa and nematodes after eating bacteria and fungi. Next, special nitrite bacteria, Nitrosomonas, convert the ammonium compounds into nitrites, NO2. A second type of bacteria, nitrate bacteria, Nitrobacter, convert the nitrites into nitrates, NO3 negative. Nitrifying bacteria do not generally like acidic environments. Their numbers, and hence the conversion of nitrogen into nitrates, therefore diminish when soil pH drops below 7. Bacterial slime, already mentioned for its ability to bind soil particles together, happens to have a pH above 7. Thus, if there are enough bacteria in an area, the slime they produce keeps the pH in their vicinity above 7, and nitrification can occur. If not, the ammonium first produced by organisms in the soil is not all converted to nitrate form. If the pH is 5 or lower, very little, if any, of the ammonium is converted. Denitrifying bacteria convert nitrogen salts back to N2, which escapes into the atmosphere. Obviously, denitrifying bacteria do not help the fertility of the soil, but they are essential in that they keep the nitrogen cycle moving. Biofilms. Bacterial slime, or biofilm, is a matrix of sugars, proteins, and DNA. The fact that bacterial slime in the soil is slightly alkaline not only influences the pH where it counts most, in the rhizosphere, but also buffers the soil in the area so the pH remains relatively constant. Some bacteria use their film as a means of transportation, literally squirting this substance as a means of propulsion. Most bacteria, however, travel using an astonishing bit of natural nanotechnology with the aid of one or more whip structures or flagella that resemble and operate like propellers. Biofilms save bacteria from desiccation as the soil dries. Soil bacteria often live inside sticky globs of biofilms, complete with an infrastructure of channels filled with water for transport of nutrients and waste. Biofilms can also be a defense against antibiotics produced by other organisms, including fellow bacteria. Bacteria colonies protected by slime are 1,000 times more resistant than individual bacteria to antibiotics and microbicides. Nutrient retention. Bacteria play a major role in plant nutrition. They lock up nutrients that might otherwise disappear as a result of leaching. They do so by ingesting them while decomposing organic matter and retaining them in their cellular structures. Since the bacteria are themselves attached to soil particles, the nutrients remain in the soil instead of being washed away, as is the case with chemical fertilizers. Indeed, these nutrients will be tied up, immobilized inside the bacteria until the bacteria are eaten and reduced to waste. Since soil bacteria don't travel very far, and there is ample source of bacterial food in the root zone, the nutrients ingested by the bacteria are kept in the vicinity of the roots. Other organisms, such as protozoa, play major roles consuming bacteria, releasing excess nitrogen as ammonium in their wastes, which are deposited in the rhizosphere right where the roots can absorb nutrients. Other benefits of soil bacteria. Some anaerobic bacteria produce alcohols that are toxic to plant life and to other bacteria. These anaerobic bacteria can be avoided when gardening by controlling the conditions that allow them to multiply: poor soil texture, lack of pore space, standing water, and compacted soil. Other bacteria are pathogens that cause disease in higher plants. The list of pathogenic bacteria is a long one, including bacteria that cause citrus canker, diseases of potatoes, melons, and cucumbers, and fire blight of pears, apples, and the like. Thousands of bacterial pathogens are in the soil, and billions of dollars are spent every year to protect crops from damage by the culprit bacteria. Agrobacterium tumefaciens causes galls or tumors to grow on the stems of certain plants. Burkholderia sapicea is a bacterium that infects and rots the roots of onions. Some Pseudomonas species cause leaf curl and black spot on tomatoes. Despite the presence of pathogenic bacteria, there are more benefits to a healthy soil bacteria population than not. For example, bacterial activity is also responsible for breaking down pollutants and toxins. These processes are usually aerobic, requiring oxygen to occur. You undoubtedly have heard of bacteria that can eat oil spilled on a beach in Alaska. There are similar bacteria that will eat gasoline spilled on your lawn, for example. Soil bacteria produce many of the medicinal antibiotics upon which we have come to depend. One can only speculate that since these bacteria have to compete not only with other bacteria for nutrients, but also with fungi and other organisms, they evolved protective capabilities. For example, Pseudomonas bacteria can correct take-all, a disastrous fungal wheat disease, by producing phenazines, very strong, broad-spectrum antibiotics. Obviously, many soil bacteria keep pathogenic bacteria in check, a big benefit of a healthy soil food web. All bacteria compete with each other and with other organisms for the finite amount of food the soil offers, and thus keep each other's populations in balance. Soils with a high diversity of bacterial types are more likely to have a larger number of non-pathogenic bacteria outcompeting pathogenic bacteria for space and nutrients. We are convinced that using the soil food web's natural defenses is the best way to keep the bad guys in check. Gardeners need to appreciate that bacteria are at the front line of defense.